Hello friends, again welcome to my channel. Now today we are discussing about one of the important concept, that is the uncoating of viral particles. Now in my previous lectures, as we are already discussing about how there is the entry of these viral particles inside the host cells, I will share the link of this video with this video. Now today we are focusing on the next step which is involved within the pathogenesis of this viral infection, which is nothing but the uncoating of viral particles. How this uncoating it is carried out. Now preferentially this uncoating, it is the process which is responsible for releasing the viral nucleic acid. As we know that the viral nucleic acid, it is encapsulated within the viral capsid. So for the purpose of the replication of these viral particles, it is important that there is the release of these viral particles within the host cell. Then and then only there is the synthesis of the viral proteins and initiation of the viral replication. So for that purpose, it is important that there is the release of this viral nucleic acid from the viral capsid, which initiate the viral replication. So the uncoating, it is a process responsible for releasing the viral nucleic acid from the viral capsid within the host cell. Now whenever these naked viruses, that is the non-enveloped viruses, these naked viruses, these are enter the cell through the process of endocytosis, and majorly the genome, it is released from the intracellular transport vesicles, or by docking at the nuclear pore complex, that is within the nucleus. As we know that the DNA containing viruses, as these are replicate within the nucleus of the host cell, and in reverse to that the RNA containing viruses, these are replicate within the cytoplasm of the host cells. Means most of these viruses, the exceptions, these are also present, that is the pox virus, which are the DNA containing viruses, but these are replicate within the cytoplasm of the host cell. In reverse to that, the influenza viruses, which are the RNA containing viruses, these are replicate within the nucleus of the host cell. Now this uncoating, it is occurs as a result of membrane fusion, which is occurs at either at the plasma membrane, means in case of the enveloped viruses. The enveloped viruses uncoating, it is carried out at as a result of the membrane fusion, which is carried out either at the plasma membrane or within the intracellular vesicles. Okay. Now firstly we are describing about the uncoating which is carried out at the plasma membrane. Now most of the enveloped viruses, those which are containing the envelope, it involves the uncoating which is carried out at the plasma membrane. That is mostly the viruses which are from the family Paramyxoviridae, such as the Sandy virus, measles virus. These are directly fuses with the plasma membrane at the neutral pH. The membrane fusion, it is carried out, that is the membrane, envelope membrane of these viruses, these are get fuses with the plasma membrane of the host cell at the neutral pH. It is said to be the uncoating which is carried out at the plasma membrane. The majorly the interaction between the viral membrane and the plasma membrane, it is carried out through the receptor ligand interaction. The receptors which are present on the surfaces of the host cell for the purpose of attachment of these viral particles. So through this interaction, the viral integral membrane protein, these are bind with the cell surface receptor. Means viral proteins, these are interact with the cell surface receptor. Means due to that, as we say that the specific type of the viruses, they can infect only the specific type of the cell lines. The reason behind that, it is majorly the presence of this receptor, because if the receptors these are present, then and then only there is the initiation of the replication cycle, because instead of that presence of that receptors, there is no binding of this viral particles with the host cell surfaces. Okay. Now the next point, it is the membrane fusion. It is stimulated by the activity of secondary viral glycoprotein. Secondary viral glycoprotein, which are called as the fusion protein. And the viral nucleocapsid, it is get released. Due to the exposure of this glycoprotein which is fuses with the plasma membrane, the nucleocapsid, it is get released within the cytoplasm. Now the F protein, which is a type of the integral glycoprotein, it is produced from the cleavage of precursor protein, from the cleavage of precursor protein, that is the F0. F0 during the transit to the cell surface, during the transit to the cell surface by the host cell protease. Means whenever these enveloped viruses, these are released from the infected cell, at that time with the help of host cell protease, there is the cleavage of this protein, and due to that there is the production of this integral protein. Okay. Now the host cell protease, these produce the two subunit, that is the F1 and F2. And both of these are held together by the means of disulphide linkage, by the means of disulphide linkage. Majorly these are 20 amino acid long, and the N-terminal domain of this F1 subunit, N-terminal domain, that is the amino terminal domain. N-terminal, it means amino terminal domain of this F1 subunit. It is highly hydrophobic, that is the water hating, hydrophobic, which produce a fusion peptide. It is responsible for producing a fusion peptide which inserts within the target membrane, that is the host cell plasma membrane, which is responsible for initiating the fusion process, Now majorly those of the cells which are lacking such type of the protease enzyme, which is essential for the production of these F proteins, majorly these are responsible for producing the non-infectious viral particles. Now, as we know that the example of the enveloped virus, that is the HIV virus, human immunodeficiency virus. This human immunodeficiency virus type first, it requires a co-receptor. As we know that the receptor for that, it is the CD4. That is majorly these are responsible for infecting the CD4 positive T helper cells. But only the receptor, it is not sufficient. Again, for the proper attachment, they are requiring the help of the another receptor, which is said to be the co-receptor. And these are the acts as an chemokine, these are acts as an co-receptor. The two major types of the co-receptors, these are present for the HIV type first, that is the CXCrd for the viral strains which are infecting the T lymphocytes, and the CCR5 for the macrophage-tropic strains of the virus which are infecting the macrophage cells. Now this attachment of the viral glycoprotein with the CD4, these are responsible for creating the high binding site on the cell surface for the CCR5. The interaction between these induces the changes in the transmembrane glycoprotein to expose the fusion peptide. Means this interaction, it is essential for the exposure of fusion peptide, because whenever the fusion peptides these are get exposed, then and then only there is the membrane fusion process it can carry out. Here it is an process. In this figure you can observe the figure how the processes of the replication cycle of the HIV viruses, these are carried out. That is firstly the attachment, then the entry, that is through the membrane fusion, there is the entry uncoating process, it is carried out. After uncoating, then these are get transported within the, it is get reverse transcriptase. Then after that the DNA molecule, it is entered within the nucleus, and then there is the integration of it with the host chromosome. Now we are switch over to the another type of the uncoating procedure, that is the uncoating during the endocytosis. Now all of the viruses, these are not uncoated at the plasma membrane. Some of the viruses, they may be uncoated during the endocytosis. Okay. Now during this uncoating during the endocytosis, it is nothing but it is an acid-catalyzed membrane fusion. It is an acid-catalyzed membrane fusion which is observed in case of the influenza viruses, which is containing the RNA, negative sense RNA, segmented form of the RNA as their genome. The entry of the viruses inside the cell, it is carried out through the clathrin-mediated endocytosis. And whenever they are reaches within the late endosome, we know that whenever through the endocytosis there is the entry, it is carried out. At that time, firstly they are transferred within the early endosome, then to the late endosome, and this late endosome, these are having the acidic pH. This acidity which is present within that endosomal membrane, it is responsible for catalyzing or enhancing the process of uncoating. The viral hemagglutinin glycoprotein, it undergo the acid-catalyzed conformational rearrangement. Acid-catalyzed conformational rearrangement which are responsible for exposing the fusion peptide. Fusion peptide. Now, majorly there is the fusion of the endosomal membrane with the viral membrane, and due to which there is the release of viral ribonucleoprotein within the cytoplasm. As we know that majorly those of the RNA viruses, these are must be associated, that is the negative sense RNA, they must be associated with the protein. So this, it is called as the viral ribonucleoprotein, VRNP. These are get released within the cytoplasm. Now this influenza virus VRNP, that is the viral ribonucleoprotein, is exposed. It's composed of the segment of RNA genome, which is surrounded by a nucleoprotein, and the viral RNA polymerase. Now the M1 protein, it is combined the viral ribonucleoprotein and the hemagglutinin and the neuraminidase proteins. These are the part of viral envelope. The disruption of interaction between the M1 and the viral ribonucleoprotein, it is essential for release of this ribonucleoprotein within the cell nucleus. Now the influenza viral envelope contains the less number of M2 protein, which is an ion channel. This M2 ion channel, it is opened by the acidic pH, which is present within the endosomal membrane, before the hemagglutinin catalyzed membrane fusion. Now due to this M2 protein, as these are acts as an ion channel, there is the entry of H plus ions, and due to this entry of H plus ions, again there is the decrease within the pH of the endosome, which leads to the conformational changes within the M1 protein, and due to that the interaction between the M1 protein and the viral ribonucleoprotein, it is get disrupted, and there is the release of this viral ribonucleoprotein within the cytoplasm. Means all of these reactions, these are carried out within the endosome. Now whenever this membrane fusion, through the membrane fusion, there is the release of this viral ribonucleoprotein within the cytoplasm, it can be imported within the cell nucleus through the nuclear import pathway. Here it is the way through which there is the uncoating, it is carried out. Firstly, the influenza viruses, through the endocytosis, they are get entered within the endosome. There is the presence of these. Then majorly with the acidification of endosome, there is the conformational changes. These are carried out within the hemagglutinin, and through that majorly the M2 ion channels, these are get activated. Again there is the acidic conditions, these are get developed, and there is the interaction between the M1 protein and the viral ribonucleoprotein, it is get disrupted, and due to which there is the release of viral ribonucleoprotein within the cytoplasm. Now the envelope proteins of the alpha viruses and the flaviviruses, these are possesses the various class of the viral fusion protein with the internal fusion peptide. Again there is the, whenever we say that there is the membrane fusion, within that there is the involvement of the fusion peptides which associated with the another viral proteins. Now within that, the proteolytic cleavage of the second protein, it excites the fusion protein to metastable state, which undergo the structural rearrangement. Again the conformational changes, these are carried out at the acidic pH to promote the fusion. And the membrane fusion of same leaky forest virus, it requires the presence of cholesterol within the cell membrane. Means each of these viruses, these are having the different mechanism for the purpose of the membrane fusion. Okay, means within the acidic conditions, again they are requiring some of the specific components for the membrane fusion. Okay. Now the another type of the uncoating procedure which is carried out within the cytoplasm, it is stimulated by the ribosomes. Means uncoating in the cytoplasm by the ribosomes. Now such type of the system, it is observed within the some type of the enveloped RNA containing viruses. That is, these are showing the pH independent mechanism of uncoating, because whenever within the endosome there is the uncoating, it is carried out, it is pH dependent uncoating. Here there is the pH independent mechanism for the uncoating. Now the capsids of same leaky forest virus, it contains a single C protein which encloses the positive strand, RNA, and is surrounded by the viral envelope, that is, which are containing the two specific type of the glycoproteins, which are nothing but the E1 and E2 protein. Now this E1 and E2 protein, the viral nucleocapsid, it contains the holes ranging from diameter 30 to 60 angstrom. Now, due to the presence of the small holes within the viral nucleocapsid, these are responsible for permitting the entry of ribosomes. We know that these ribosomes, these are of smaller size. So these ribosomes, these are get entry inside this viral nucleocapsid. And the capsid, within the capsid, there is the beginning for the process of translation of the positive sense RNA, and the nucleocapsid, it must be disassembled. Due to the translation process, there is the disassembly of this nucleocapsid, which mediate through the interaction of ribosomes with the C protein. Means whenever these ribosomes, these are entered within the nucleocapsids, these are interact with this C protein, and this C, due to the breaking of the interaction between the C protein with the nucleocapsid, there is the disassembly of the nucleocapsid. It is carried out due to the presence of these ribosomes. Okay. Now the another mechanism, it is the uncoating, which is carried out by the disrupting the endosomal membrane. Disruption of endosomal membrane. As the different pathway that we are studying, within that we are describing, that is there is the membrane fusion. Now here there is the uncoating, it is carried out by disrupting the endosomal membrane. Now the adenoviruses, these are entering the membrane through receptor mediated endocytosis. These viral particles, these are transported via the endosome from the cell surface towards the nuclear membrane. As the adenovirus, we know that these are the DNA containing viruses. So these are go towards the nuclear membrane because these are replicate within the nucleus of the host cell. It undergoes the sequential steps for the uncoating, through which the structural protein, one by one the structural proteins, these are get released from the adenovirus. The viral capsid, it is get destabilized due to the acidic pH of the endosome. The structural protein V causes the disruption of endosomal membrane and deliver the remainder of the particle, that is the nucleocapsid, within the cytoplasm. Then this subviral particle docks onto the nuclear membrane. It docks onto the nuclear membrane, and the different viruses showed the variation within their modes of the uncoating depending upon their morphology and the cellular structure. On the basis of their morphology, their genome structure, the different types of the proteins which are associated with these viruses, on that basis there are the different mechanisms of the uncoating. The small size poliovirus, they are release their nucleoprotein by forming a pores within the endosomal membrane. The naked icosahedral orthoreovirus, they are containing the double stranded RNA as the genome, which are uncoated within the lysosomes. So here there is the information about the uncoating of the viruses. So as if you like this video, please subscribe my channel and also share this video with your friends. Thank you.